In this video, we'll continue our class on gastrulation, and basically, we'll be focusing on gastrulation in Amohioxus. But before we talk about the gastrulation in Amohioxus, let us talk about the cleavage in Amohioxus first. Let's just brief ourselves about the cleavage in Amohioxus. Now, we have to understand that the cleavage in Amohioxus lanceolatum is holoblastic. So they undergo what we refer to as holoblastic cleavage, also referred to as complete cleavage. You understand? Now, this cleavage it involved the complete division of the cells into from 1 to 2 cells referred to as the blastome, from 2 blastome to 4 blastome, from 4 blastome to 8 blastome, from 8 blastome to 16 blastome. Now, during the beginning of this particular process of cleavage, you will notice that the cells of each of the blastome are of equal sizes. Look at this one, equal size, compared to this one, equal size. Now, when we reach the 8 cell stage, what we notice is that the cells that are at the upper surface are becoming smaller, while the cell at the lower surface are becoming bigger. You understand? Like the size of the cells, not the size of the zygote or the embryo itself, just the size of the individual cells. And at the end of the gastrulation, there is a formation of cavity, and this cavity is what we refer to as what? The blastocell. You understand? We refer to this cavity as the blastocell. And we notice that there are now cells that are lining the what? That are lining the side. And these cells that are lining the side of the blastula, because this is what we refer to as the blastula. So this is the blastula. You now see that the cells that are lining the blastula, they are small cells, something like this. They are very, very small. But the cells at this place, they are large. Same as here. Yeah, same as this one. You see that these cells are small compared to the cells that are here. You understand? You now see the parts that are having small cells are what we refer to as the animal pole. And the parts that are having large cells are what we refer to as what? The vegetal pole. Which means in this case of Amohioxus, because yolk are evenly distributed, we cannot determine the animal pole and the vegetal pole based on the number of yolk. Rather, we are going to determine the animal pole and the vegetal pole based on the size of cells at the end of blastulation or at the end of cleavage. And the animal pole are the one that tends to have the smaller cells. The vegetal pole are the ones that tends to have the larger cells. You understand? So in the blastula of Amohioxus, there are areas that can be distinguished. For example, we have the vegetal pole with larger cells. We have the animal hemisphere or the animal poles with smaller cells, and we have the marginal zone. That is what they are trying to show here, the marginal zone. Now, what we notice is that you see this animal pole are those ones that contain the presumptive skin epidermis and the presumptive nervous system. You understand? Like presumptive, or possibly, and that is what we mean by presumptive. You now see the cells at the vegetal pole. They are referred to as they are the presumptive endodermal cells. Are you getting me now? The presumptive endodermal cells. And the marginal zone or the marginal zone are the one that contain the presumptive mesodermal crescents. You understand? And the area for the notochord on this particular side. So what are the things that we are going to distinguish? So this is everything that this particular write-up is trying to say. The cleavage is holoblastic. After the 8 cell stage, you will see that the cells are having different sizes, and the cells at the upper parts, which is having smaller cells, are referred to as the animal pole, and the one at the lower parts, which is referred to as the vegetal pole, and there's formation of cavity we refer to as blastocell. That is basically what cleavage in Amohioxus lanceolatum is all about. You understand? Now, let's now talk about the gastrulation in Amohioxus itself. I won't really show right up in this particular part. I just want us to understand this concept. So I'll be using different images. I have this image here. I have this old structure here. And I have this one here as well. You understand? This one is showing something like a fate map or something like that. And it's showing the onset of gastrulation, the early gastrulation, the mid gastrulation, the late gastrulation, and the cross section of gastrulation. You understand? Now, let's now talk about this. Now, the first thing, the gastrulation is actually initiated by the blastoderm at the vegetal pole becoming flattened. You understand? That is the what initiates gastrulation itself. Flattening of the cells at the vegetal pole. You understand? That is the first thing that happens. And you will see that the shape of this particular structure is round, Abi. And you will notice that the cells that are at the animal pole are smaller than the cells at the vegetal pole. Now, the first thing that happens is that if you look at it very well, this one is curved. But during gastrulation, the first thing that happens is the flattening of the cells. You understand? Now, after the flattening of the cells, there will now be invagination. Remember, I told you that invagination is the inward movement of cells. That's why I said we are going to be talking about the times that I explained the other time. So there will, let me be writing it down. The first thing that happens is what? Is flattening of cells at the vegetal pole, right? Flattening of cells at the vegetal pole. I won't be wasting much of our time. Vegetal pole, right? Now, the next thing is now what? Is now infolding of cells, you understand, to form a cup shaped structure. You understand? It forms a cup shaped structure with a large cavity. So there will now be infolding of cells. You see that the first thing that happens is flattening of cells. The cell becomes flattening. And if you look at this image C very well, you see that they are going inside small, small. They are going inside small, small. And at the end, they have moved inside completely, right? They have moved inside completely. You see this particular thing that happened here, that is what we refer to as what? As invagination, infolding of cells to form a cup-shaped structure with a large cavity which open with the exterior on the side that was originally the vegetal pole of the embryo. You understand? You know that this particular side is originally the vegetal pole of the embryo, right? Now it is now forming a cupped shape structure. This cupped shape structure is now open to the exterior, like the outer surface of the embryo, from a cavity that is previously the vegetal pole. Remember that this place is previously the vegetal pole. It is now a cavity now after the cupped shape structure has been formed. Do you understand? Now, what is now going to happen? You see, this cup-shaped structure is going to have a double wall, right? And what are these double wall? That is the ectoderm. Look at it. This is the ectoderm and the endoderm, right? So let me clean this so that we can see it very well. It is going to have double wall, double wall. That is the ectoderm, the ones that are lining, and the endoderm, the one that are inside. You see that we haven't talked about the mesodermal layer yet, right? We have not talked about it yet because the mesodermal layer is also at outside. It is located at the outside. It does not move inside. And that is where the concept of involution and ingression comes in. We are going to talk about that. So the it has double structure. That is the ectoderm and the endoderm. Now, we now notice that this was our blastocele, right? This is our blastocele. This is our blastocele. This is our blastocele. What is now going to happen is that there will now be reduction in the size of the blastocele. Because if you look at it very well, if you look at this blastocele very well, it is something that is very, very large before, but it is becoming smaller as we are forming the cup shape. It is becoming smaller as we are forming the cup shape. It is becoming smaller as we are forming the cup shape to form a very small one. And you will later see it that it will later become obliterated at the end of gastrulation. You understand? So you see in this stage, there is still a space between the external and the internal. You understand? There is still a space between the external and the internal. And that space between the external and the internal are the remnants of the blastocele. And I'm going to show you what the remnant of the blastocele looks like now. Now look at it. I want to clean this so that we can see it very well. I hope you're understanding. So this is the blastocele. You will notice that this blastocele, the color of the blastocele is light pink. And the color of this particular place is what is peach. You see that it is not present here initially. So this is the new cavity that will be formed. We later call it achentheron, don't worry, or the primitive gut. And this one that is light peach is the blastocele. So whenever you see this one, just know that it is the new cavity that is formed due to gastrulation. And this one is the what? Is the blastocele initially. So what is going to happen is that as this thing is moving inward, what is going to happen? They will be occupying the space. You understand? They will be occupying the initial space of the blastocele. You understand? They will be occupying the initial space of the blastocele. So they are moving inward. They are moving inward. Can you see? This is supposed to be the blastocele. This lining is supposed to be the blastocele. This lining that is here is supposed to be the blastocele. You now see that we now have a very small remnant of the blastocele that was present initially due to the what? Due to the infolding. You understand? Due to the infolding of cells during gastrulation. Now, the external lining, which is the ectoderm, which is the external lining, contain of the presumptive skin and the presumptive nervous system. So let me just write it somewhere. So I will say ectoderm consists of what? The presumptive skin and the presumptive nervous system. I'll just use NS to it. Now, it consists of the part which has been classified as the ectoderm. You understand? So that is the reason why we call it ectoderm. The external lining called the ectoderm. The internal lining now consists of the presumptive gut material, that is the endoderm. So the reason why we call this particular part endoderm is because it consists of cells, the presumptive cells of the gut. And remember, I have already mentioned initially that the gut is formed by the what? Is formed by the endoderm. You understand? Now, the presumptive mesoderm cells and the presumptive notochord is now found within this particular space called the mesoderm. You understand? So everything we have been explaining since morning now is just invagination. You understand? Invagination. So after the invagination, we now have the ectoderm and the endoderm only. The mesoderm still lies outside. We don't know whether it's going to go into the middle or anywhere. You understand? But for now, you have to understand that the ectoderm, how can we determine the ectoderm, that is the external lining, and to be carrying the preservative cell of the skin and the nervous system? How do we determine the endoderm? It will be inside and it will be carrying the presumptive gut material. How do we determine the mesoderm? It will be at the outer layer, but it will be carrying the cells of the presumptive mesoderm and the presumptive notochord. Do you understand? Now see the presumptive material of the mesoderm firstly lie at the rim of the cup shaped. You understand? It firstly lies at the rim of the cup shape. You see that it is lying at the rim of the cup shape. Can you see? It is lying at the rim of the cup shape, at the outer layer of the cup shape. But very soon, they will now move inward. That is what we refer to as what? Inward movement of cells, also known as the involution. You understand? Inward movement of cells. So they are going to move inward to occupy a space in the internal layer of the cells. And I'll be showing you that because that was not shown very well in this particular part. So I'll be showing you that in this. Okay, let me use this structure. So this structure, you see that the first thing that happens is the flattening. The second thing that happens is invagination. You see, invagination, they have invaginated, invagination. And if you look at it very well, we now have, I want to look for something. I'm looking for something. I'm looking for the cells of the mesoderm. They will be at the rim of this cup. I don't know if it was indicated. It was not really indicated, but it was indicated at the end of this particular place. But how can we determine it? Look at these cells. Look at the shape of these cells. Can you see the shape of these cells? You will see that they are the ones here, right? They are the ones at the rim, right? They are the someone at the rim of this particular place. You understand? Now, you now see the the mesoderm, they are now going to move from the external wall of the cup into the what? To occupy a space in the interior. You understand? Which means at this point, the external layer now consists of only the ectoderm at this point. When the mesoderm now move inside, the external layer now consists of only the ectoderm. You understand? It only consists of only the ectoderm. And the inner layer now has the what? Now has the endoderm. And between the external layer and the internal layer, we now have the what? We now have the we have the mesoderm and the what? And the notochord. You see at this stage that the ectoderm has been formed, the endoderm is also there, and the mesoderm has also been formed. We can now call the structure, we can now call it gastrula now. You understand? You now see the movement, the inward movement and the infolding or the inward bending of the endoderm and the mesoderm is also referred to as invagination. You understand? It is what we refer to as invagination. Now, during invagination, there will now be formation of cavity. The formation of what? The formation of cavity. And this cavity is what we refer to as what? What we refer to as the arc enteron or the primitive gut. If you look at it very well, the invagination, right? The invagination here give rise to formation of cavity. Cavity means something like an empty space. Look at it. The cavity that we have here, that is what they refer to as the gastrocele. You know that we call the one for blastula blastocele, and we call the one for gastrula, we call it gastrocele. But in actual sense, we call it the arc enteron. After it has been fully formed, we call it the arc enteron. Now, you see the arc enteron is opening to the exterior. It is opening to the exterior through a particular space called the blastopore. So, what is happening here? Invagination leads to the formation of cavity. This cavity is what we refer to as the arc enteron. The arc enteron is open to the external surface called the what? Called the blastopore. You understand? So, the blastopore denotes the pathway by which the endoderm and the mesoderm passes to the interior. You understand? The blastopore, this particular blastopore, it denotes the pathway at which the mesodermal cells that are here, that is the mesodermal cells that are here. You know that this is the ectodermal cells, right? This is the endodermal cells, and we have the mesodermal cells here. So this blastopore refers to a space at which these mesodermal cells now move inward so that they can occupy their space. Do you understand? Now, the rim which surround the blastopore of the cup-shaped embryo may likely to be the mouth. This rim that we are talking about here, this rim here is likely to be the mouth, and that is the reason why they call them the deuterostome, because they form the presumptive mouth first. The protosome rather, protosome. If it was the deuterostome, it would be the anus that will be formed first. You understand? So it will be the mouth, and it is usual to speak of the lips, of to call it the lips. You understand? Now, we may distinguish the dorsal lip from the ventral lip because there will be formation of lip. We can may distinguish the dorsal lip from the ventral lip from the lateral lip of the blastopore respectively. You understand? The blastopore is very broad in the initial state of gastrulation, but very soon it becomes like it contracts. You understand? It contracts into the ankenteron, which makes it to reduce to a very smaller size when it compares to its original orifice. Because if you look at it very well, in the initial stage, we have a very large blastopore, right? We have a very large blastopore. But as we are going, the blastopore will be continue to become smaller until it is reduced completely. You understand? And when it is now reduced completely, we are now going to be having formation of lips. And these lips are the dorsal lip, the ventral lip, and the lateral lip. We also have the lateral lip as well. So it's supposed to be like indicated out here. So we have the ventral lip, the dorsal lip, and the lateral lip. And very soon, the blastopore becomes obliterated completely, and we're going to be having a gastrula. You understand? Now, this happens, cells continue to move inside. So as more material is shifted into the interior, the interior of the remnant of the blastocele becomes obliterated completely. You understand? The blastocele that was here initially. Remember, I told you that we have remnant of the blastocele here. Very soon, as cells move inward, they becomes obliterated completely, that they won't be there. You understand? Blastocele becomes obliterated. So the presumptive notochord and the mesodermal crescent shift into the interior of the gastrula. I've mentioned that before. And they also change their position relative to each other, like mesoderm and the distin, because the mesoderm and the notochord, they lie on the opposite side of the embryo. You understand? They lie on the opposite side of the embryo. And if you look at it very well, we have them here. So if you look at it, we have the what? We have the notochord here, and we have the mesoderm here. You understand? So they like now the lateral horns of the mesodermal crescent converge towards the dorsal side of the embryo, and they came to lie on the both sides of the presumptive notochord. Now, in the next stage that follows after the contraction of the rim of the blastopore, remember I told you that there will be contraction of the blastopore, and in this contraction of the blastopore, there will later be complete obliteration of the blastopore itself, because blastopore will be reduced to a very smaller size. You understand? Now, what is now going to happen after that is the elongation of the notochord itself. There will be elongation of the notochordal cells. Understand? There will be elongation of the notochordal cells and the mesodermal material. You understand? Now, when they are elongated, the external wall of the gastrula, it takes part in this elongation as well, because it is not only the internal part that is going to elongate, the external part is going to elongate as well. And one of the results of this is that the presumptive material of the nervous system will become stretched. That is what they refer to as the neural ectoderm here. You understand? We call it the neural ectoderm. It will now become stretched into a longitudinal band of cells lying mediodorsally, like in the middle, but at the dorsal part. Look at it. It is at the middle, but at the dorsal part of the embryo. It is lying mediodorsally over the notochordal material, and it is being broadened than the notochordal material. You understand? So what you are going to see here is just like a rearrangement. What is going to happen is that there will be elongation. You understand? Elongation of cells. And during this elongation of cells, there will be elongation of the ectodermal cells as well. And the elongation of the ectodermal cells sees the increase or the elongation of the neural ectoderm, or the presumptive neural material. If you look at very well, this one is very, very small, and this one is becoming what? Is becoming elongated, is becoming large. You understand? And this neural ectoderm is seen to occupy the mediodorsal layer. You understand? When you say middle, that is the middle, like the middle part of the embryo. And when you say those are at the back, you understand, towards the back of the embryo. And it is lying over the what? Over the notochordal material, because this is the notochordal material. You understand? So this is the end stage of gastrulation. Now, this gastrulation is now driven by a conserved but a simple set of molecular pathways that establish the body axis, they specify the germ layers, they coordinate the cell movements such as invagination, involution, convergence, and extension. The there are there are key molecular pathway. You understand? The key signaling pathway like nodal or lefty, activin pathway like TGF, beta family, WNT, Catherine signaling, and fibroblast growth factor pathway. These are molecular distinct that initiate the formation of gastrula during the gastrulation of Amohioxus. So this is where we'll be stopping today, and I hope you understand the gastrulation in Amohioxus now. In the next video, we'll be talking about the gastrulation in amphibians, which you have the slide ready, and that is what we'll be talking about in the next video. I hope you understand what I explained here, and this will actually do justice to what we have explained before. This is what I'm trying to say. The neural plate, which is the presumptive nervous system, it is now lying mediodorsally towards the what? On the notochord. You understand? And if you look at it very well, there is extension. You understand? So this is extension so that they can form a what? A round structure, something like this. And there is extension. So this is basically what gastrulation is.